Hello, everyone. My name is Chris Cepeda, and I am a rising senior at Stanford and a Way Fellow for the summer. Today, I'll be talking to you about my project for the summer, which was designing mRNA vaccine constructs against HIV-1. The HIV epidemic has been addressed for decades, and the problem is that we still do not have an effective vaccine for it. One of the reasons we still do not have a vaccine is that HIV-1 has a rapid mutation rate, and this results in enormous viral diversity. Thus, it is hard to build a vaccine against all of these strains. There is a solution to this problem. Broadly neutralizing antibodies, also known as bNAbs, have been discovered and that target conserved epitope regions on HIV-1 ONV. If we could elicit these bNAbs with a vaccine, we could get protection against a wide range of HIV strains. This solution has a problem, though, because these bNAbs have extensive somatic hypermutations, so researchers have struggled to elicit these bNAbs through conventional vaccine strategies. Given the excitement about mRNA vaccines with SARS-CoV-2, we were interested in exploring whether the mRNA vaccine technology could be applied to eliciting bNAbs because they potentially mimic natural infections more accurately. This method is more accurate because there is prolonged expression of the antigen, so you could elicit more bNAbs. However, the problem with making an mRNA vaccine for HIV is that HIV-1 ONV has poor expression when compared to SARS-CoV-2 spike. From there, our approach became, how can we improve expression of HIV-1 ONV for the development of an mRNA vaccine that produces bNAbs? We could try two things. We could try to get rid of the two endocytosis motifs from the native cytoplasmic tail, and we can add several stabilizing mutations. On the left shows a mutation researchers like to call SOCIP. In its entirety, the image on the left shows the surface protein HIV-1 ONV. These mutations stabilize the surface protein, which tends to be very flexible due to HIV-1 ONV being able to undergo a lot of conformational changes. This flexibility is part of what makes HIV-1 ONV expression so bad. So stabilizing ONV through mutations such as SOCIP, MD39, and DS increases expression. On the right shows a mutation called I-short. This mutation gets rid of two endocytosis motifs, first by mutating a tyrosine to an isoleucine, and then truncating the tail to remove the endocytosis motif on the end. Here are the three main methods I used. On the top, we have the Gibson assembly. In the middle, we have a shortened version of the tons of mini preps I've done this summer. And lastly is the seeding, transfecting, and flow cytometry experiments I ran. For the flow cytometry experiments, we used VRC34 as our primary antibody, which mainly targets the fusion peptide within GP41, which was key because the GP41 remained the same throughout the strains we tested. Then we used a fluorescently labeled secondary antibody that binds to our human IgG VRC34. So in these figures, the further the orange line is to the right compared to the control, the better the expression of the surface protein. As you can see from these first two figures, the expression of SARS-CoV-2 spike, shown on the left, is much better than the surface expression of HIV-1 ONV, shown on the right. As HIV-1 ONV is nearly identical to the control, you can see that there's poor expression. In the figure below, we show our discovery that expression improves with stabilizing mutations. We have not been able to test the stabilizing mutations individually yet, but we now know that having MD39, DS, and SOCIP works together really well. We also found that removing the two endocytosis motifs with the I-short mutation had little effect on improving HIV-1 ONV expression. We also looked at the germline targeting immunogens to see how they expressed, and we found that RC1, which elicits antibodies for the V3 epitope, expresses similarly well to BG505, a different strain. The RC1 3-fill variant adds glycans to part of ONV that we do not want immune responses against, and this expresses slightly worse. Then we looked at IgG2, which elicits antibodies targeting the CD4 binding site, and the triple MIC, which elicits antibodies against targeting 3 different epitopes, and they all express fine. From this experiment, we have concluded that adding stabilizing mutations enhances expression of HIV1-ONV, and truncating the endocytosis motifs had little effect on improving surface protein expression. Future directions of this project include testing the stabilizing mutations individually, synthesizing mRNA, testing those for expression in vitro, and if that looks good, packaging them into lipid nanoparticles and injecting them into mice to measure immune responses in vivo. To end, here are my references and acknowledgements. Thank you for listening.